Just send me to. I know where to be.
All right, everybody, we're going to get started here. I've got 8:30 on my end, and just to stay mindful of everybody's time, we'll get going right away. So thank you guys for coming. I know there's going to be a few more people, I think, trickling in here through the morning. So I think we should have enough chairs, and then we were joined by quite a few online as well. So I've got a few different professionals here today to talk about highly pathogenic avian influenza, or better known as bird flu. We have Dr. Heidi Pecoraro, we've got Dr. Jake Galbraith, we've got Dr. Logan Wiend and Ryan Herigstead, Levi Schlosser, and Crystalline Coons. Everybody's going to take a little bit of info on all of this morning and present their side of things and how they look at it from their perspectives and in their fields. We're going to do a little bit of hands-on as well today, and then hopefully you guys can take some things back and use this in your guys' communities. I kind of decided to do this. This was put on at a public health conference earlier this year, the North Dakota Public Health Conference, as one of our sessions. NDSU kind of had already did some of the work and put together the speakers. So I took some of those same speakers, brought them here, and then added a couple additional ones to it as well. And then we are going to do. I added a little bit of a bonus topic regarding the New World screwworm, as that's the thing that's up and coming. I've been watching that for a while as it's come up through South America and through Central America. So pretty nasty stuff. This has been an ongoing outbreak for a while now of this H5N1. So I thought it would be very timely, especially during the fair with all of the producers and stuff that are in town, and try to catch a few people to spread some education. So it's prevalent to us and very important to us in public health to do this education as well, because it is something that we are monitoring in case it does mutate into a disease that will increase transmission through humans. There have been a couple people in the United States that have acquired this disease, but it hasn't made that genetic mutation yet to turn into like a pandemic level type of thing. So we're just going to start here and do some education to work on our prevention and try to spread this through the community a little bit. So I appreciate you guys all being here. Those of you in person, the bathrooms are just straight out through this door and in the lobby. Our exits are here and here. If you guys need anything, obviously let me know. And then there are coffee, snacks, and stuff up there for you guys. So we go until about noon today. If we go a little over, that's fine as well. But I want to make sure you guys can get some lunch and things too. So starting there, we have Dr. Pecoraro, NDSU. I'll let her introduce herself and give her info right now. So you guys, it's all you, Heidi.
Great. Just getting it pulled up here. Started on my last slide, so let me just pull back up to my first slide. And share. Hopefully it works. Fingers crossed.
Yes.
Perfect. Does everyone see it?
Yep, we're seeing it.
Fabulous. Okay.
So,
Okay, here we are. So I'm going to talk a little bit about a couple emerging diseases in the U.S. livestock that Zachary already alluded to: avian influenza and New World screwworm. Again, I'm Heidi Pecoraro. I am a veterinarian, and right now I'm the attending veterinarian at NDSU. I'm actually trained as a veterinary pathologist, and for 7 years before becoming a attending veterinarian, I was at the veterinary diagnostic lab working as a pathologist. 4 of those years, I was a director. In addition to being a pathologist, my PhD is actually in influenza, believe it or not. That's probably why I'm here. And specifically in how influenza jumps from one species to another. And I use canine and equine influenza in a lot of the things that I worked on. Our roadmap for today is I am going to talk a little bit about the virology of high-path avian influenza, how it's transmitted among animals, some of the clinical signs, and then again, just touch on New World screwworm, what it is, and some of the mitigation efforts. A big thing to note is, we're going to talk about livestock, but these diseases that the focus of today is, they're actually, they're from other animals. They're diseases that livestock get from other animals. So with high-path avian influenza, It's influenza viruses typically come from wild birds, waterfowl, or domesticated poultry. And with New World screwworm, it's not a worm. It's actually the bot or the larvae of a fly. So we'll start off with high path avian influenza. And I have a couple illustrations coming up, and I not to overwhelm you, just to give everybody a little bit of background on the virus. I'm not trying to make anyone a virologist, but just so that we're all talking about the same things today. With influenza, because this is from the virus side of things. With influenza, we have these two big concepts, and I'm sure you all have heard of it, an antigenic drift and an antigenic shift. And again, it comes down to the virus itself here. And so this right here is like a virion. It's the structure of the virus. And on the outside surface, we have a couple of proteins, the hemagglutinin, neuraminidase. So that's where we get our H. So H5N1, the H is hemagglutinin, the N is neuraminidase. And then inside the virion, we have 8 gene segments. Now, this influenza is what we call a negative sense single-stranded RNA virus. It's a lot to say. It has to do with the genomic structure, and then also where and how it replicates. The important point is to know that because it's a single-stranded RNA virus, how it replicates, there's a lot of mutation that happens with influenza viruses. So with antigenic shift, we have these small mutations over time, and it changes what the genome encodes for like 11 proteins. It changes the structure of some of these proteins, or the makeup of some of these proteins. So if you had influenza virus in the past, or had been vaccinated, well, your antibodies that you had to fight off the previous influenza viruses aren't going to work when they're, against changes to these proteins. And so over time, the virus changes, and that's why we have to get a flu shot every year, right? But when we're talking about antigenic shift, it's much more dramatic, much more drastic. So the segmented genome, can actually, if you have the right species and the right cell type, an animal can be, including humans, can be infected with several influenza viruses at once. And if it's in a host cell and all the conditions are correct, right for this, the segments can actually reassort. So you could get genome segments from one virus into another, and you can make up a whole new virus. And it's often in these cases that we see pandemics or huge outbreaks. So not only is there there's absolutely no prior immunity to these types of viruses. So that's from the virus side of things. I want to just touch on a little bit about the host side of things. And again, not to make you guys virologists, but just a schematic, we have a lot of different kinds of influenza viruses, and they're typically species specific. However, all of those, like equine, canine, swine flus, those originally did come from avian influenzas. Various mutations occurred where then they become much more species specific. And so, horses have their own type that they spread amongst themselves, and dogs. And in fact, equine influenza is actually what mutated to become canine influenza. One of the main points, though, is that, a virus isn't going to be able to infect a host unless the host has the proper type of receptors for that virus. And in our own upper respiratory and lower respiratory system, we have a compilation or different types of receptors, more concentrated on one type of receptor in the upper versus the lower, and those different kinds of receptors are more or less susceptible to becoming infected with different influenza viruses. And I'm not going to touch on this because it's just so much to talk about. I want to just quickly touch on the fact that, I was talking about these different segments, genomic segments. Well, there have been research that showed that each mutations in each one of these segments can actually affect how a host cell can be more or less susceptible to a virus. So it's not just the the hemagglutinin or neuraminidase that's important. All of these gene segments can make a difference in terms of whether or not a virus can enter a host cell or not. And then finally, getting back to agriculture and livestock. If you follow this illustration, influenza viruses circulate in wildlife. Typically, they're circulating all the time. Typically, doesn't cause much disease at all. It can be persistent in the environment, and we see some spillover of some of these avian influenza viruses into our livestock due to feces or environmental contamination, fomites, etc. And then occasionally, some of those, influenza viruses can actually spill over into humans too. Again, typically not causing very much disease. It is often in this agricultural setting where we will see some major mutations in influenza viruses that cause sporadic and sometimes devastating disease and mortality. And that's when we often see these high-path avian influenza viruses. Now, these high-path avian influenza viruses then make their way back into the wildlife population where it can cause havoc, and then these migratory birds, right, often, and they can spread it all over the world as we've seen. And then we can also see these high-path avian influenza viruses in wet markets, but also in poultry workers. And later we'll talk a little bit about cattle, dairy workers as well. So agriculture does play a role in this whole cycle of avian influenza viruses getting into our livestock. So especially pigs are one of these animals that can harbor various influenza viruses, including some from birds and humans, and they're called the great mixing vessel. And they can, after a big reassortment, it can be a brand new virus that can, wreak havoc all over the world. Okay, so just a quick touch on this low path versus high path avian influenza concept. These low path avian influenzas circulate in birds. There's usually no, in wildlife, waterfowl specifically, typically no clinical signs, maybe mild disease, usually self-limiting. And that's pretty much due to the fact that in these low path avian influenzas affect the intestinal tract where the receptors are for these viruses. And then they're shed and might be environmentally persistent, but the virus itself does not go beyond the mucosal layer, the epithelial tissues of the gut. When we're talking about high path avian influenza, it's a totally different story. It can actually affect the gut too. That this illustration isn't 100% correct, but it does also infect the respiratory tract. From the respiratory tract, it can go into the bloodstream, become systemic. Respiratory distress is a major sign that we see, a clinical sign that we see, but also just sudden death. Like you'll just go in and find a bunch of birds dead into a birdhouse. There's virus replication all over. It goes past the epithelial mucosal barriers to organs, skeletal muscle, and including the central nervous system. And what's interesting is high-path avian influenzas often do show as neurologic disease in animals that, especially mammals, and I'll talk about that in just a second. Often in like birdhouses, they die so quick before we even see neurologic disease, but it definitely can happen in birds too. So again, sometimes we'll see neurologic disease without any other clinical respiratory disease signs at all. High path tends to be more neuropathogenic, so it likes the nervous system, it likes the brain, especially cranial nerves and the olfactory nerve. It's very important route of infection into the brain. And then once it's in the brain, it can affect all different kinds of cells in nervous tissue cells. Here's a picture of different mammals throughout the United States that have been confirmed to have high path. These are wild and captive mammals. Typically in these mammals, the clinical sign was acting weird, like neurologic disease. And for the most part, I would assume, especially especially the ones that we've done here in North Dakota, our first thought is that these animals have rabies. They're mammals, they're acting neurologic. And so they're euthanized or they die, and the brains are sent to us. We do rabies tests first. We get a negative. We look under the scope at the tissues and we see inflammation. So our next thought is perhaps this is influenza. And these are such cases. You see, we have birds, marine mammals, various captive, domestic, exotic cats, even lagomorph, raccoons, skunks, all different mammals have been infected with high-path avian influenza. And the thought is that these guys, I told you about the olfactory nerve, that they're probably scavenging on wild birds or potentially domestic poultry. And while they're scavenging, their face is down there, they get a nice shot of the virus right up into the olfactory nerve that goes right into the brain. So that's mammals, and we've seen a lot of neurologic disease, but a couple of years ago, we started seeing something different, specifically in dairy cattle. And we weren't seeing respiratory disease. We weren't seeing neurologic disease. Instead, there was these herds that had, some dehydration, maybe some mild respiratory signs. They were off food. They had maybe a fever early in the course of the disease. But the biggest clinical sign was that they had a decrease in milk production. And what milk that they had was nasty. So, this yellow, chunky, sometimes red, curdled milk. And speaking of raw milk, no one would ever want to drink this. But this is just, just gross milk. And then what would happen is that, this was, for the most part, self-limiting in that the cattle would survive. However, they never really came back into milk production, at least great milk production. And eventually figured this out, that it was high-path avian influenza. And as a pathologist, I think about in terms of what the tissues look like. And these tissues under the microscope, the glands are just wiped out. There's tons of inflammatory cells. There's a lot of necrosis or dead cells. And when these animals recover, what would replace all that dead tissue was fibrosis. And so these animals never really came back into milk very, very well. And so they would be pulled that way because of those concerns. I'm just going to give you the high points about this study. It's one that I was on with Dr. Webb at the diagnostic lab here. We've been working with some researchers at University of Pittsburgh on receptor type of studies. And so these are some pictures of some dairy cattle mammary tissue. Here's a gland in here and here. And then all these blue areas, a lot of inflammation there. Well, here's a trachea. This is the lining of the trachea. This is a mucosal surface. These are all epithelial cells. Same with what lines the mammary gland. The point of this one is, if we go back here, they've done studies to highlight where the receptors are, if they're a human type of receptor or a bird type of receptor. And what they found is in the mammary tissue that they have an abundance of both type of receptors for both birds and human type flus. But in the trachea, they didn't have that so much. So it's one clue on why maybe it's actually probably why dairy cattle, their main clinical sign is mastitis rather than respiratory disease. Shifting real over to North Dakota, because we're here in North Dakota. Let's talk about what we see here. I got these data in May from the lab, and over the time, so when I first became the director, it was, I think, 2022, and I believe it was pretty much right after that we got, like, within months that we had this outbreak. So talk about fire by baptism, or baptism by fire, other way. We started getting, we started really quickly, we got on board to be able to do the testing, because these are all regulatory tests. Once we were on board, we started testing, and since that time, we've done over 200 tests. We can pool samples, so we can have up to 5 birds in one test. So this represents over 1,000 birds, and 75 of those samples were positive. Now, it's important to know that we're going to test where we suspect disease. So This isn't just like thinking that 30% of all of our poultry have a high path avian influenza. This was very specific testing. In terms of the cattle that we've tested, 2 years ago, I think it's been now, when we found out that dairy cattle had high path, then there were various regulations in place in terms of testing milk before animals were able to be moved. They've done about 200 tests representing over 800 animals, and there have been no positive samples in North Dakota in the milk here. But we have had some mammals, non-bovid mammals, so not cattle, where we have suspected it, mostly foxes and cats, and of that we've had 5 positive samples. Here's another paper that Dr. Webb and I are on, came out of our lab, and so these are from some cats, some barn cats that came to our lab that had high path avian influenza, which was not suspected at first. So one of the things that the lab does is, a big thing that we do is investigating cause of death. And so these cats were sent into us that had a little bit of respiratory disease, but were mostly neurologic. And so, of course, with respiratory and neurologic disease, they were worried. It would be weird I think there were 5 cats and we got 3 of them. It would be weird for it to be all rabies, but precautions were taken when they did the autopsies. And so rabies testing was done, some other testing was done. And when the tissues came back, this is the lung tissue, and these are alveoli, and mostly alveoli should be open or filled with debris and inflammatory cells. And this is some cerebrum, so part of the brain, and this is a blood vessel that is surrounded by inflammatory cells. So both pneumonia and respiratory disease is what we saw. Eventually, we got permission to test for high path avian influenza, and it came back positive. These were animals that had access. I guess they were scavenging off of wild birds, but potentially one of the people in the home also had been on dairy farms. An example that hits a little closer to home, as in just right up north from where I'm at. This is north of campus here in Fargo. Off of 19th, we have a number of barns, animal barns. We have a beef barn up here. We have a sheep barn here, and a dairy barn. Over here is a lagoon. I had gotten a call from someone out of the sheep barn that there was a Canada goose that was acting weird. And when I got on site, the goose was tilting its neck back. So that's a sign of neurologic diseases in this kind of animal, and birds where their necks tilted back. And I was like, oh no, that's high path avian influenza. And so I said, let's not touch it. Let me get back to the wildlife, game and fish people here in the state and see what we should do. And so that's what we did. And then I had heard from wildlife, okay, we're sending you samples. So I was like, oh, great. Someone came out, took care of the bird. I'm getting those samples. We get the samples. We test them. They come back. We say non-negative. We say not detected. So it's negative. And I'm like, okay, well, that's interesting. We'll see if there's any more animals that die. And then I did hear about, this one did die. And then I heard about 2 more dead ones out by the lagoon. And so I talked to the barn manager, and he says, like, no, actually, no one came for the birds. That one's dead out front, and then there's ones in the back. So by the lagoon. So I went out, put all my PPE on, triple bagged them, took them back to the lab, got permission to test from Game and Fish, and they said yes. And again, it came back not detected, so negative. Now, any animal like this, we are not going to do that has high suspicion for high path. We are not going to do an autopsy on, but because we're having this die-off, we got permission to go ahead and do the autopsy. So Dr. Mitchell did that, and we did it in a hood, even though they're huge birds, but in abundance of caution. And what she found in the pancreas, this is the pancreas here. This is the duodenum. The duodenum is the top portion of the small intestine. In the pancreas, we had all these spots. So these spots represent dead tissue. And as a pathologist, my 2 differentials are, well, this could be Newcastle, but most likely this is still high path. It's like, this is high path. And especially if I have these dead birds everywhere, I highly doubt we're having a Newcastle. It's possible. I was like, these guys have high path. So even though it came back not detected for us, we sent it off to the samples, swabs off to the national lab, and they had to do a couple things, and it did come back positive. So the good thing is that we picked up these birds. There was no signs in any of our dairy cattle or in the sheep, but you can imagine we were concerned to have high paths so close to our dairy barn. Oh, the other birds from Game and Fish were from another part of Fargo. They were having another die-off, but assumptions when you don't get clarification. I thought they were our same birds. Let's talk really quickly about people as well. There has been spillover, especially in dairy, from dairy herds and poultry farms. 71 cases to date. I think someone else can touch on this, but just to say that, the people that often get this are those that are working closely with these animals. Okay, I just got a couple more minutes. I'll take questions right after these slides if we do have time. But just to touch a little bit on a new world screwworm. Again, it's from a fly. Up to this, I pulled these data off just last week, July 14th. We're up to 39 cases. These are mostly in Texas and New Mexico. And you can see that it is mammals here, but we've seen it in sheep, goats, cattle, dogs. The screwworm can infect any mammal, basically, any warm-blooded animal. So just to talk about that a little bit in terms of their life cycle, typically it's like 21 days. It depends on temperature, anywhere from 18 to 24 days, depending on how hot it is outside. So again, it's from a fly, an adult blowfly. They will lay 200 to 400 eggs along the edge of a wound. Then those eggs, the larvae hatch within a day. And what's interesting is they feed off of living tissue. They don't go to the dead tissue like a lot of other maggots would do, but they live off of they eat off of the living tissue. So you can imagine that there is just tons of destruction in that wound. And that the necrotic tissue that they're creating and all the decay attracts other flies. And so then more flies come and lay more of their eggs along the wound's edge, and it just perpetuates the cycle. Those larvae eventually fall off and then pupate into the soil and emerge as an adult. It's really important to note that this is a painful process to the animals that get this disease, because again, this is living tissue. And from what I read, death is typically 7 to 14 days in animals that are not treated. I really like this pamphlet that American Livestock Markets and Dealers Association put out, and this is on the web. You can find this because it shows you what the fly looks like. And again, these are flies, so they're 2-winged. You shouldn't have 4 wings. They have 2 wings, and they have this kind of rusty orange eyes. And this is the side of a wound. Sorry, anybody, turn away if this is too much for you. But they like to lay their egg packets right along the side of the wound. Right here, you can see, and right here too. And what kind of wounds do they like? Well, they like the umbilical cords. They like castration sites, branding sites, ear tags, ear notching and marking, the genitalia, dehorning sites, and basically any cuts or scrapes or open wounds. So these are the types of things that you should look for. There are some points from the state vet that he's been putting out, and I touched base with him last week. He talked about some of this. He does want people to know that this does not have an impact on food safety. It's not infectious. It can't be passed from animal to animal or from animals to people. Of course, you have to think about a fly being an animal, but that's a different story. That's not what he means. And this is preventable and treatable with proper management, and if you can recognize the signs. So how do we treat and mitigate this? Again, you have to recognize it to be able to report it. But even if you are just thinking, this looks like this looks like a weird wound, or even a wound, you'd want to report that to your veterinarian. You can debride these wounds and apply topical sprays on livestock to get them through it. In our companion animals, we have more tools in our kit, some medications, some insecticides that we can give them. But the cornerstone of the efforts to get rid of New World screwworm really relies on sterile insect techniques. This is how we approached it decades ago and was the reason why we were able to eradicate it the first time. And so what is a sterile insect technique? Basically, in a lab facility, you would rear or breed a bunch of these blowflies. Then you separate the males and the females, and the males undergo this gamma radiation, and that makes them sterile. Then those sterile flies are released over towns and cities so that they can compete with the wild sterile males, fertile males, and outcompete them with the ladies. So then the ladies then will lay infertile eggs, and then slowly it gets rid of those flies. And this is just my last slide to talk about really quickly how important biosecurity is in both these situations, especially high path. Dr. Galbraith is going to pick it up from here, but there are things that you can do on farm to mitigate these, to stop it before you even have a problem. Okay, that was a lot of talking, so I'm going to stop sharing and see if we have time for questions.
All right.
Can you see me?
Thank you. Yep,
I
we
lost
can
you.
still see
Okay,
you,
good.
Heidi.
Okay.
Yeah. Thank you very much for that presentation. Does anybody in the room have questions? Anybody online? We have a couple minutes here if anybody has anything. Personally, I think the New World screwworm thing is pretty gross. I've seen some photos of it online, of what it does to humans and stuff as well, and they do leave a pretty nasty mark. So I do appreciate Dr. Pecoraro talking a little bit more on that, as that's kind of been up and coming here these last couple of years as it's moved up through the South. But yeah, really, really great information. I do have, oh, okay. Yeah, so I do have one question here in the room. I'll see if you can hear her, Heidi. Let's try it. Go ahead, Lisa.
Is there any prevention for livestock producers for high path?
So for the New World screwworm or for high path?
I was learning about high path, but.
Okay, so there have been some studies on vaccination. I don't know if Dr. Galbraith's going to talk about that at all, but I do know there have been efforts for vaccination. And in terms of New World screwworm, you know, fly control is very important.
Thank you.
Okay, I'll ask. For the screwworm, weird name, the countries to the south of us where it's originating from, I'll say, are they doing anything to mitigate this?
Okay, this is not my area. However, I have read that there are facilities that do the same thing, trying to do the sterile insect technique as well.
Tom, I've got some information I have prepared on that too. So,
Okay.
Great. Any other questions? Okay. Well, thank you very much, Heidi. Again, it was really great information. I do have a 10-minute break built in. I do have coffee and snacks and stuff over there, and some handouts and things if you guys want to grab on all these topics that we're talking about today. So we will meet back here at about, let's say 9:12. All right, can everybody online, can you guys see the presentation and stuff here?
Yep, thank you.
Okay, awesome. Thank you very much. All right, so we've got Dr. Jake here. He's with NDSU Extension. He's going to do some more on high path AI, and really give some more, delve into a lot more information in regard to it. So we'll reconvene now, and then he does go for a little bit here with his presentation, and then we'll break again for our next bit. So I'll let you guys know. So just be ready for that. Obviously, you can step out if you have to or anything. So all right, I'll turn it over to Jake.
thank you. I'll try to just go a little bit with my presentation, not a too much bit with my presentation. So, I'm Dr. Jake Galbraith, the extension veterinarian at NDSU. I've been in this position for just over a year. In the decade or so prior to that, I was in private practice, what we would consider what we call a mixed animal practitioner, so seeing both livestock as well as small animal, companion animal. Most of my trade was in cow-calf work, so probably about 75% cow-calf, and then 20% dog and cat, 5% grab bag, whatever else walks through the door or calls up on the phone. So yeah, going to talk a little bit more about HPAI, some background on the current, kind of a timeline of the current outbreak as it pertains to livestock. I'm also going to talk about a joint project that we've been having over the last year and a half between NDSU Extension, Animal Science, North Dakota Health and Human Services, and the Center for Immunization Research and Education. I think I got that acronym right, CIRE. So we've been doing a cooperative project on that for, it was just for 2025, but we got the funding that was left over extended to be able to use it yet in 2026. So that's one of the reasons why I'm actually in town here today. I passed out some pamphlet brochures to the people who are present here. We just got those hot off the press on Thursday afternoon. Not soon enough for NDSU Communications to put them through their automatic folders. So if you notice that the folding lines are a little bit askew, that's me. You should see me try to wrap up Christmas presents. It's pretty ugly. So. All right, I'm going to try not to go over too much ground that Dr. Pecoraro already covered. She did a great job describing the disease, the virus of itself, as well as how that's affecting animals. So I got a lot of pictures in here, a lot of diagrams. So just kind of recapping the current outbreak. This is of course a diagram showing some of the different clades of virus across kind of the migratory pathways. I really like this diagram. It shows the common migratory waterfowl pathways as they cross our continent, and really seeing these arrows, especially that red arrow, how that crosses right over North Dakota. We're in a major migratory flyway, and how these lines intersect, of course, coming over from from Europe and Asia. And so you can really see how when we have an infected population on one continent, how these lines will then crisscross each other, so we can see exposure that will get into the other migratory waterfowl patterns. And then also this map shows the deeper red colored states are where we have much more poultry production going on. I think the deep reds, it says there like 770 million to 1.4 billion numbers of poultry sold on a per year basis. And so when we see those flyways going over those areas, it really brings into focus just how serious this outbreak has been and has continued to be. So this really started, first detected this strain in 2020 through Europe, Africa, the Middle East, and Asia, primarily seeing them in wild birds. We did see this migrate into to Europe, over into Canada, and then the first detection in the U.S. took place in 2022. In 2022, of course, when it broke out here, it kind of came with a little bit of a vengeance. Had the first outbreak in January in wild birds, and then just a month later, we saw it in a commercial turkey operation. Not too long after that, saw the first human case in the U.S. from a poultry worker working in close contact, of course. And then throughout the next few months, we, referencing back to that map that Dr. Pecoraro showed across the United States, seen it in various mammal species as well. When 2022 broke out, so I'm also part of what's called the Veterinary Reserve Corps. So it's a group of private veterinarians across the state who the state can contact in cases of things like this, where they need someone to be able to go and do some sample collections on behalf of the state. And during 2022, I did get called out 3 different times to do some investigations. So that was kind of a new experience for me, and really, made me think more about my biosecurity things, putting on my PPE just to make sure that I'm not spreading things from an infected farm to the clinic, and of course also take things home. When this happened, this was kind of before we really had seen some of the feline cases, and I got a couple of cats at home. And so once that was kind of figured out, I kind of was thinking, okay, if I have to do this again, what am I going to do to make sure that I don't take this home to my pets? And we've had some information that we go out about that to the public as well. But in my situation, be like, I'm going to make sure that I take completely separate clothes, wash everything up at the clinic, bag it up before I bring it home. I've even joked about it. Well, I'll just get the garden hose out in the backyard if it's a nice day, and like what you see on TV when they're decontaminating people with the mops and everything. So So this is just a list of the North Dakota cases in 2022. So it's a pretty extensive list. I tallied these up off of the website just a couple of days ago. So the total number of birds affected in North Dakota in 2022, almost 250,000. And so this is going to include both birds that were infected by the disease, but also ones that had to be depopulated because of a positive case. And what happens in those situations is if there would be a positive case, either in a commercial operation or in a backyard, private operation, they get the positive result, and then those animals are depopulated. Unfortunately, just to try to help prevent that further spread around the state. When we would do these investigations, we would collect the samples, but we would also do an inventory of the number of live birds that were on the premise, because there was some indemnity being paid out by the federal government at that time. So that way, if they got a positive result, they could then be recompensed for the birds that would have to be eliminated. So moving to 2023, a little bit of a drop down in the number of cases, but still a pretty high number of total overall birds affected, almost 150,000. So from 2022 to 2026, nationwide, we've seen over 200 million losses in the commercial poultry industry. That's just commercial operations. And actually, as of right now, we're at about 208, 209 million based off of the USDA website. And then most of the birds that are affected in commercial operations are the egg layers. And so, when we had some of these issues with egg prices, 2-3 years ago going through the roof, of course, now we're hearing some other information about maybe some collusion within the egg laying industry that might have been going on at that time. But also, I think there was some supply and demand issues going on in response to avian influenza. And it's not terribly surprising that egg layers would be more affected as a commercial species, because when we think about what an egg layer is doing, they're going to be laying eggs, so they're going to be in these facilities for a longer period of time, as opposed to like broiler chickens who are just being raised for meat. They're going to be all in, all out. You can really kind of tighten up that biosecurity pretty well, because you don't have to be going in and out, taking out eggs, putting in eggs. And these animals, once the entire barn full of broilers is done, you can take the entire coop out, do a very thorough cleaning job inside those facilities. Whereas in the egg laying situation, they're in there for a much larger or a much longer period of time. You've got so many more inputs going in and out. So there's more chances, not only on a time scale of exposure, but also on a person interactive fomite potential during that time. So that's why we would see more egg laying operations being affected by this. So 2024, that's when things got kind of more interesting. And, I'm sure it's the same in human medicine, but when you hear a veterinarian say, ooh, things are getting interesting, that probably isn't very good news, unfortunately. So I was in private practice this time, and I remember this March goat case showing up from North Carolina, a positive case of HPAI in this goat on a farm with positive poultry. I remember that story coming out and just going, that's interesting. We don't usually see influenza crossing into our ruminant species, goats, sheep, cattle. It's just not something that is usually on our radar. And so I was like, okay, that's pretty interesting. Same time, we were dealing with a quote unquote mystery illness in Kansas and Texas that was causing upwards of 20% reductions in milk production in these dairy operations. Personally, when this came out, I was kind of thinking, I bet there's maybe some smoke inhalation, because there was some very massive wildfires taking place in Oklahoma at this time. I thought, well, maybe there's some air quality issues that's affecting these dairy barns, and I was totally wrong on that, of course, because then we found out that it was HPAI affecting these cattle, and that really was very interesting to all of us. And so, April of 2024, we saw the first cow to human transmission. Pretty understandable to see why those things would happen, because when you look at a commercial milking parlor, the workers are they're below where the cattle are standing. They're about eye level with those udders, putting on the pumps and everything. And so any splashing that takes place, whether it's from the milk, which can be very, very virus laden, but also we can see virus discharge in urine. And of course, cattle, they're not going to be shy about where they're going to be doing their eliminations. And so get splashing up, gets into the eyes, just like with a normal flu virus. Don't touch your eyes, don't rub your eyes. When you got something that's splashed up in your eyes coming out of the south end of a cow, you probably want to wipe it out pretty quick. And so that's probably where we saw most of those exposures. And there's things we can do to of course, address that: face shields, gloving, of course. And then, after we found this out, there was mandatory testing for interstate travel. So any lactating dairy cows that were going to cross state lines had to have a negative test for HPAI. So again, this is back into North Dakota cases in 2024, kind of a lighter year for us, fortunately. Whether this is just because of more awareness and stricter biosecurity practices, or just we got lucky with the migratory season, tough to say for sure. 2025 kind of had a little bit of a rebound here nationally. Another 50-some million poultry and upland game birds were affected. Ohio got really smacked. Almost 15 million laying hens were killed or had to be depopulated in Ohio. And this is just a screenshot of the USDA's tracking page for HPAI. And this, you can go through this and look at state by state, for whichever year, whichever month. It's really interesting. It's kind of fun to play around with, but you can really kind of go down the rabbit hole with it. You look at like North Dakota, we've had probably around 500,000 birds affected if you were to hover over this on the whole the length of the outbreak. Minnesota, I think it's somewhere around like 30 million birds have been affected by this outbreak. And then you look at like Hawaii. I think Hawaii there was like 2 cases affecting 5 birds total. Puerto Rico, I think, was 1 case affecting 1 backyard bird. And so it's really interesting just to see how, you're definitely going to know which states have got poultry production in them based off of this map. And then moving into 2025, kind of, again, that rebound year, saw some more breaks. And this was kind of interesting last year with kind of the timing of some of these. So typically we see our outbreaks during the migratory season. So in the spring, when the geese are moving up north through North Dakota, and then again in the fall, when they're heading back down south. So last year, our fall outbreaks really extended, quite late into the season. So through December, and actually we had our last one in January of this year. And so that was very surprising to see those outbreaks that late in the season. But of course, if you drove around, you could see some geese, out on some open water in that time frame too. And then this year so far. Like I said, we had that one outbreak in January. Had our typical ones we're going to see here in the springtime. So far, knock on wood, it seems like it's been a little bit of a lighter season. But another couple of months, we're going to start putting that to the test for the fall migration. Getting into the cow side of things, Dr. Becker had a lot of this picture up here already, so not going to rehash too much in terms of covered ground, but things we would see in dairy cattle. So mild serous drainage from the nose, some mild depression, lethargy, mild fever, just kind of like what you would expect person to be with a mild flu. But again, really a high affinity for that mammary tissue causes some pretty significant mastitis. Like she said, you don't want to drink that stuff even if it were pasteurized. But definitely, I'm not going to get into the politics of raw milk consumption at all. That would be a whole knockdown, drag out kind of an argument probably. Map of where we see that tissue being affected, of course, and then also kind of a timeline of when we expect to see virus and antibody production on a time scale here. Of course, can affect the respiratory system, but again, just that high affinity for mammary tissue. And again, not a surprise why we see pretty drastic reduction in milk production, like I said, upwards of 20%. And in a 5,000 head dairy, maybe seeing upwards of a 16% reduction across an entire herd. That's a lot of money. That is a major economic impact. And then this is just based off of the North Dakota animal import regulations. So this was put into place. Basically, it's the same thing. Any lactating dairy animal that would come into the state needs to have a negative HPAI test before they can come in. This has been in place for probably over a year at this point. And so just wanted to highlight that is continuing on. So moving into the project that we've been doing so far this past year. So some of the activities that we did. This project is really designed to try to get outreach and exposure to, for the most part, people who are involved with backyard poultry operations, commercial poultry operations. Not only talking about educating them on what the disease is, what the virus is, how we can try to take steps to limit our exposure, limit spreading it to our pets, to our livestock, our backyard flocks, our commercial flocks, but also protecting ourselves, and then really trying to educate people on viral reassortment and the importance that regular seasonal flu vaccines does play in trying to help prevent reassortment and development of a more serious strain of this virus. And so last year, so I joined NDSU in June of 2025. So we started out basically about this time last year, had posters up at the state fair in the poultry barns regarding general hygiene practices. Had conversations with exhibitors there as well. Moving into the Big Iron Farm Show in Fargo, I think that's August or early September. Again, had another poster display there, mostly talking about reassortment and the importance of vaccination. Was able to do an in-person presentation at Big Iron as well, and then also an interview with Ag Week talking about, again, this project and the importance of vaccination. Moved on into the fall, we have an annual event at NDSU called Moose, Use, and More. It's basically a showcase from the animal science department where the researchers can show some of the work that they do, some of the research they do. The different animal units will have livestock there for people to interact with. It's basically just a big show and tell. We usually get around 1,000 kids and adults who show up, really geared a lot towards kids. But last year we wanted to have this project involved in it as well. So talking about hand washing, talking about how we can prevent, spreading the flu to ourselves. So we had some hand washing stations that we rented. That was the first time we actually got these hand washing stations for Moose, Use, and More. And one of the biggest feedbacks we got about, because I was one of the co-chairs at this event, one of the biggest feedbacks we got on this event was, we love the hand washing stations. We just wish there were more of them. So we learned something as an event that people want stations there and having those demonstrations, because we also had demonstrations using glow germ where we would have people put the glow germ on, go do a hand wash and come back and see how good a job they did. And especially with the kids, of course, they love that. They got the glow germ on them. It's like, ooh, look at this. It's really neat. So I think it really does a great job of trying to hammer home good practices and get people thinking about the things that they need to do in order to help protect themselves. Stockton Convention last fall in September, I did a presentation that included talk about this project, and then also at the Walsh County Fair in Park River, had some handouts up there, talk with poultry exhibitors as well, and then kind of been continuing in that line a lot in the last 6 months. That's a blank slide. Oh, that's it. Yeah, I got animations of this thing too. I forget about that sometimes. So this is at Moose, Use, and More. This is the glow germ and washing station. Just that's a kid who's being able to look to see how good of a job he did. Then we had some coloring books there to engage with those kids as well. And this is where we had those hand washing stations. We had 4 of them. So this year we're probably going to get 6 or maybe 8, so that we can station them at other areas, because we weren't really thinking about it, but really, if you're going to go and be petting a lot of sheep and pigs and reaching into a cannulated steer, even though you got gloves on, people might want to wash their hands a little bit before they go get a hot dog or a bottled water or something. So it was a good lesson for us to learn. And then last fall, or actually winter, I guess, December, first part of January, I did some in-person HPAI presentations in 4 different towns across the state. Basically, these presentations would involve description of the influenza virus, much like Dr. Pecoraro did, discussing reassortment, the importance of vaccination, talking about the history of the outbreak, like I just went over. We did some biosecurity and PPE demonstrations. Also did a biosecurity risk and reward game that was developed by University of Minnesota Extension. I have those materials here. Thought about having it like a little break right here to do that, but the people who are tuning in remotely wouldn't get very much out of that, and it might kind of slow things down. So if there's any interest in going through a round of it, it's about 15 minutes. We can do that after everybody's done. It's just a little demonstration, mostly geared towards like 4-Hers, FFAers, about how to not bring nasty things home from the fair, things like that. Then, of course, we did some glow germ projects where we had kids and adults, practice putting on PPE, go and handle some stuffed chickens that they didn't know had glow germ on them, and then practice taking off their PPE. Oh, you didn't do a very good job of not touching your face there, did you? So again, trying to hammer home those lessons. So through these activities, utilizing some of the CDC handouts that we have, just trying to show how people are going to be picking up HPAI potentially, aerosolized dust, foam mites, things like that. Same thing in the dairy industry, the splashing, the liquids that are going to be potentially getting on people, getting in their faces. And then this is the one that we had in, trying to remember which town. Once I get the map, I'll be able to see it. But again, having some of the people try on these PPE, practice doing a line of separation where you put on your PPE, step over a line, whether it's even just whether it's a bench or even just a piece of tape on the floor, having to practice those things. Other things that we did is try to get outreach to hunters, because since this is in our migratory populations, unfortunately, the first case that I went to visit back in 2022, 2023 to do an investigation and sample collection. We're pretty sure that the way that they got HPAI was that the son was out goose hunting. He brought the goose back home into the yard, the field dressing, or not field dressing, but to dress the bird in the yard. And then the chickens on the premise had access to the same location. And we're pretty sure that's where they got exposed to. But of course, it could even be that they flew over and pooped in midair and got the exposure that way. So we did, this is just a picture. You can obviously see this could be a major potential for spreading disease somewhere else. Not to mention, hopefully there's more than one person involved here, or else we could get Game and Fish involved with a little bit of poaching potentially. So we did release through extension some materials to try to get the word out to hunters that we should try to do field dressing as much as possible, so we avoid bringing those things back home. If we do have a pickup that looks like this, once you're done field dressing, take that through the car wash. We don't want to have this running through your yard, especially if you work with chickens, work with dairy operations. Is there something coming up here says I can still see it?
So when you say field dress, you have to have some kind of idea on that bird to bring back home though.
Mm-hmm.
You can't just breast it out in the field and just bring the breasts home. So what do you suggest people do for that?
I guess I would have to defer that answer to the Game and Fish department because I've never been a waterfowler. Do they have leg bands or anything you can place on them to identify them that way? I've only ever done pheasant hunting where you just leave one leg on to prove that it's a rooster, of course. Right. Yeah. So I don't know.
Yeah, you still have to do the same thing with waterfowl.
Yeah. Biggest thing on that is just don't bring them home to do it. And, whether you have to go to a different station to do this and take care of that. But yeah, I guess the specifics on that I'd have to defer to Game and Fish. I went through that. So we had the 4 meeting. Sharon, that's the first meeting we had. I was thinking of Finley first, but Sharon's right down the road. So we had 4 of these meetings. We were planning on having 6. One got canceled in Williston due to the blizzard that showed up that night, which was great because I had already gotten out there and got stuck out there for an extra day. And then one in Minot, it was going to be that next day as well. So between weather and just everything else going on, this was in December. So of course, there's lots of holiday activities going on. But we did have good attendance at the 4 meetings that we did have. And then really overall, the reviews that we got back on this were very positive, very appreciative of the information that we were distributing out there. People really liked the fact that we were describing why vaccination is an important part of this. And, they like being able to interact with PPE. I had a table very similar to what you've got over there with just PPE stacked to the gills for people to, even if they just wanted to try on, different glove sizes so they know what size of glove fits them. There's a lot of people out there who just don't know what size of glove they have. And so that can be really important as well. These are just some more feedback from the I guess feedback things that we ask people to fill out. And then of course we've also developed quite a few different handout materials. Like I said, we got the brochure that I passed out in here. So now we're over 30 education materials. We also have a what's it called? An online. I'm not technical by any words in terms of like computer technology. It's like a shared drive or something like that, so that people can access it through a QR code and get more information. Like the QR codes are on the pamphlet right there. And then of course done a series of interviews throughout the last year about HPAI. When I'm sure when fall comes up, we have some outbreaks that occur. I'll probably get called up by Ag Week or Prairie Public, do some interviews again about that. And always, whenever I do these, I try to tie in back to biosecurity, PPE, vaccination as well. So our plan for going forward through 2026, we have the funding extended until January 27th. So developing more materials, doing more of these meetings, hopefully get some more of those lined up for this fall. I still plan on going to 2 or 3 more county fairs in the state to be there when they're having their poultry exhibition, so we can be able to answer questions, but also just get some good information out to them. We're not going to do the game right here since we talked about that as far as being a little bit of a time thing, and the people online really wouldn't get anything out of it. But I do have some more information on world screwworm. I guess before I get into that, any questions on the HPAI, the outbreak timeline, activities we've been doing through that project? Yes, ma'am. Usually the recommendation is like 2 to 3 weeks. Yeah, and I think I have that in the pamphlet there as one of the recommended guidelines that, you bring things home. And it's not just with poultry, but also it's a good idea with any cattle, sheep, goats. So you put in there that both on your herd and your flock, it's a good idea to quarantine because you never know what nasty little bugs you might have brought home. Just as a quick aside, a personal story on that. So I grew up in Oaks, and we raised Suffolk sheep. And my oldest brother, this is like 10 or 11 years ago, so just 2 years into me being a vet. So my oldest brother, his kids, took a couple of market lambs from the home flock and showed them at shows throughout the summertime, and then brought them back kind of late August, because they were done with the show season, and brought them back. And about 3 to 5 days later, we had one of the lambs, not one of the show lambs, but one of the lambs they were put in with, just up and died. Hadn't shown any symptoms or anything, hadn't shown any signs of illness, just was dead. It's like, oh, shoot, well, that stinks because this was a pretty good sized lamb. And the next day there was another one. And the next day another one. And the next day, I went and I did the chores and had to pull out the fourth one and looked at everybody, see anything looking else that was wrong. And about 3 hours later, my brother comes in and he starts chewing my hide. Why didn't you pull out that dead lamb? I did it this morning. Well, there's one sitting out there now. So we had to go through and do, a mass medication, metaphylaxis. So we gave everybody a shot of antibiotics. Basically, this was a Haemophilus infection in the lungs. And so it would hit them hard, it hit them fast, and they would die very quickly. So we learned a very unpleasant lesson. We lost 6 lambs out of that whole ordeal. Luckily, the antibiotic metaphylaxis stopped things. But that was a hard lesson to learn, but it's one that I've really kind of taken, and it's one that I can share with people to be like, this is real, this is legit, you don't want to mess around with this. Quarantine is important. Sorry, I think you had your hand raised too?
Not to belabor the milk issue at all, but I know this is transferred through inhalation. Can the virus be acquired through ingestion?
Yes, I believe that it can. I don't know that if they've identified it happening in humans directly, but we have seen cases of it in cats, barn cats who were fed milk that was infected and, showing up with, pretty serious neurologic impact. So we know that it can be through ingestion as well. Yeah. And we'd also see that with that mammal map, a lot of the mammals that were shown on there are scavengers. And so, if we have a goose that dies and a fox or a coyote or a cat or whatever comes up and eats on those, that has been a demonstrated line of transmission as well. Yeah. Any other questions before I move into graphic pictures ahead? So I did this presentation first in January. So this was before it got to not to North Dakota, thank God, got to the US. And so have had these materials. I've updated some of it a little bit. The first one I like to always talk about is just the name of screwworm, the Greek name. It is very apt because this is a creature that we have known about for hundreds of years. So Cochleomyia hominovorax. So cochleо in Greek means spiral or snail or a screw. Maya means fly. Hominis is human. Vorare is to devour. So a fly that devours humans in a spiral or screw-like manner. It is a very appropriately named pestilence, put it that way. Dr. Becker already covered, how these vermin affect animals. We've got these pictures on them. You look at those mouth parts. Oh, I'm looking at the next one ahead. Look at these mouth parts. That is just a vile-looking creature. You think about how much that's going to hurt going through. When they talk about debriding the affected tissues and spraying them, it's really recommended to also give pain relief to these animals because once you start trying to pull these things out, they will try to burrow in deeper to get away from whatever you're trying to treat them with, and that's going to cause quite a bit of pain to those animals. Just some more kind of on-file pictures. Again, we've covered this territory already, the life cycle. One of the nice things I always talk about when people ask me about this is we don't really have to worry about a population getting established up here because it does not survive in soil temps less than 46 degrees Fahrenheit. So winter is good for something. There is one good thing that we can like about winter is that it keeps things like this away from us. The only really threat that we have for getting New World screwworm up here is for it to be carried up here on an animal or God forbid, a person, and that would be, potential that then you could maybe get a localized outbreak, but it's not going to be something that can persist, throughout the season. More fun pictures. This is, I think, one of those key Florida Keys deer from what was that 2016. I have it on the map up here in a little bit, I think. But that was the last outbreak we had in the U.S. In those Florida Keys deer. So going into treatment options. So we do have some products that are under conditional approved and also emergency approved. There are no fully approved medications that are for New World screwworm. And I say medications because there is an insecticide that we use in livestock called Catron 4 or Catron IV. It's a spray that we use very extensively, like when we would do dehorning, castration, you spray that onto a wound. Mostly we would do it to prevent other types of flies from having fly strike in those areas. But that one is approved by the EPA for treatment of New World screwworms, but it's an insecticide, not a medication, so it's in a different ball of wax. So on these conditionally approved products, we have Dectomax injectable, which is, it's a macrocytic lactone, or what sometimes you'll hear people say, it's one of the ivermectins. Ivermectin is the original one that came out with Dectomax. We got Doramectin. There's all these different, there's Sidectin, there's all these ectins out there, but people will just say, oh, it's just one of the ivermectins. So if I say that, that's what I mean. It's in the ivermectin class. So the injectable Dectomax, that is a product that is over the counter. It's been available. We use it for internal and somewhat on some external parasites in cattle production. We've had that for 35, 40 years at this point. The next one is called Exolt. Maybe I'll just talk about that when I actually have them brought up there. So talking about how these products can come to be approved. So it kind of breaks it down into different classifications. A fully approved medication, the manufacturer has to prove that the drug is safe when used according to the label directions, and there is substantial evidence that it is effective for the intended use. So we don't have any products on the medication side that meet that requirement for New World screwworm, mostly because this isn't a problem we've had to deal with in the last 50 years, basically. So there really is no financial incentive for manufacturers to go through the laborious and expensive label changing process to make to have a product labeled for something that basically no one's been seen in the last 40 or 50 years. It's somewhat understandable. So we have conditionally approved medications. So the bar that these ones need to cross is that the drug is safe, again, when used according to the label. There is evidence for reasonable expectation of effectiveness for the new indication, which would be screwworm. And they have to recertify for that every 5 years with showing that they're making active progress in gathering evidence towards approval. So they can get this conditional approval. They can re-up it every year for up to 5 years, but they need to show that they are making progress on trying to get towards fully approved status. And then we come into emergency use authorized medications. These can only be used in response to a public health crisis, not a livestock crisis, not an animal health crisis, a public health crisis. And so since screwworm can affect humans, that's why this crisis was declared, I think it was back in like a year ago or so. So the known and potential benefits have to outweigh the known and potential risks. And then this is only allowed for the duration of the emergency. Once that emergency is passed, then you can't use these products in that way anymore. So fully approved medication, 0, Catron 4, again, that's an EPA thing. Conditionally approved, we have the Dectomax, the Exolt, and also Credelio, which is a dog and cat medication. And then we have a number of these emergency use authorizations. So this is a chart off of the FDA website showing these products on here. So it's hard to see it on the screen here, but you see Dectomax, it's got that CA1, conditionally approval 1, which is on all 3 of those conditionally approved products. You'll also see Dectomax in the emergency use authorization. So that's to allow it to be used on dairy cattle as well as sheep, goats, and horses. So since Dectomax on its normal label is not approved for use in those species or for that class of cattle, in order to be used on them, now they have to have an emergency use authorization. So that's why we see it on 2 lists. This is a lot of fun information when you're trying to talk about this and assuage some of what the product reps are talking about as well. So this is Exolt. This is a pour-on product. This is brand new, basically, for use in cattle. This is originally developed to treat mites in poultry. And then just in the last year and a half or 2, they developed a pour-on or the same product for cattle. I believe it's also kind of a little bit of a response to some of the ticks that we've been seeing out on the East Coast that are carrying filariosis. Big thing with this one, 98-day withdrawal period. So if you're going to use this, you're going to be married to those animals for for, 3.5, 4 months. Dosing prescription only. You can't get this over the counter, as opposed to the Dectomax injectable. This is for treatment and prevention. Again, this has been something that has been available for producers for decades. Does have a, about a month withdrawal period, but again, this is available over the counter. It does have a label indication for that it should prevent reinfestation for 21 days. That I put in here, not all ivermectins are created equal. And one of the things that goes on with this is that there's lots of different forms of ivermectin treatments in livestock. So this is an injectable. There's porons, there's oral medications. Not all are going to be working the same way. So with injectable, ivermectin products, that will also work on blood-sucking lice because the animals, they're on the outside of the animal, they suck blood. You get higher plasma levels of that ivermectin with an injectable. So it'll work on those ones that suck blood. If you use a poron product, you're not going to get those same high levels of ivermectin or whatever mectin we're talking about in the bloodstream, and so it won't work on those blood suckers. Now, we don't know right now whether or not poron of an ivermectin is going to have sufficient levels to kill New World screwworm, or whether an oral medication will. But what I can see potentially going forward is that producers who might not be well informed about this are going to say, oh, my cattle are okay because I poured them all with generic ivermectin, poron. I hit them all in the alleyway. We're going to be okay. That could be where I could potentially see the possibility of failure of treatment and prevention. So it's just one of the challenges that we see, especially in livestock medicine, where, especially with ivermectin, we've all gone through the COVID years. We all heard how much ivermectin was either just the miracle drug or it was worthless. As with all things, it's probably somewhere in between. But it's just like when you say pandemic about HPAI viruses, if you talk about ivermectin, right now they've all got political undertones to them. So I could see where this could be something for some difficulties discussing with some producers. Okay, history lessons. We already talked about how we've done eradication in the past. Production facilities, Dr. Pecoraro talked about, we're ramping up that production. So the one that we've had for the last 20 some years in Panama is called COPEG, and that has the ability to produce 100 million sterile flies a week. They are retrofitting a fruit fly facility in Matapa, Mexico that should by the end of this summer get up to 100 million per week. So that brings us up to 200 million. It took 500 million per week during the original eradication efforts to get this out of the US, down through Mexico, down out of Central America. So we need to ramp up that production. We've got that coming online in Texas. It's going to be 1.5 years, 2 years yet before that gets to the full productivity. So we're going to be fighting this bug on a seasonal basis for probably the next 2 or 3 years before we really start to make massive progress. So right now, the nice thing is that, we're getting past the fly season even down in Texas. And so hopefully we'll be able to take the existing sterile flies that we have, maybe be able to push that back down a little bit further south during the wintertime months. And then it's just, hopefully we can get it a little bit better under control as more production comes online. This is the picture of that COPEG facility in Panama, very picturesque. This is the Matapa one in Mexico, again, pretty picturesque area. And this is the only picture I could find of the Moore Air Force Base back in like the 1950s. This is where they actually did the loading up onto the sterile flies onto the planes to do the aerial spraying there. And so, yeah. This is a map from the 80s that shows the historical range that these flies could potentially go as far north on a natural basis. Again, if this was to get on a steer that was heading from Texas up into Nebraska or South Dakota to go to a feedlot and it had world squirm, all bets are off at that point. The nice part is you're not going to get an extensive population develop because winter is going to kill them. So this map just shows the history of the eradication efforts in the United States. Oh, one thing with that Panama COPEG facility, so those 100 million flies that we get per week, the deal is with that is that, so we were trying to keep that from coming up into the US, but if it does come to the US, as a country, we get all of the flies. We get first pick at all of the flies in order to control things here versus down there, which really stinks for them because they are in a hurt unit down in Central America. Yes, you have a question? The sterile flies? That is a very good question. I would probably refer back to the life cycle, but probably I would say just like 5 to 10 days, something like that. I don't know for sure, but I may be just pulling something out of my ear there. And again, so the eradication effort started in the 60s, moved down to Mexico through the 80s, 2000s, finally got it down to that Darien Gap where Panama meets Colombia. It's a natural kind of barrier because it's just so much jungle. Unfortunately, through the movements of people, cartel movements, taking illicit animals across borders, that's the big suspicion that we have right now of how that has moved up north. Just another map that shows this, but also shows that outbreak in the Florida Keys in 2016 and the deer population there. And so then we move into current status. So this is a map that I really like. This is from just over a year ago. And so it's hard to see on the map. You have a close-up that you can see. So the numbers, they show, when it's going up through these countries in Central America on a year-to-year basis. So it starts in July of 2023 down in Panama. We go to number 2, that's in July of 2023. We get up into Honduras in September of 2024. So it's just a steady march that has been coming up over the past 3 years. So last year at this time, we were hoping, do I have a mouse? Yeah. We were hoping that we could get it stuck here at the neck of Mexico by doing sterile fly bombardment in this area. That was the hope. Unfortunately, as we all know, that did not occur. I think weather patterns had something to do with that, but as well, when we're talking about 100 million flies that were designed to work down here, and then you try to apply that same number in a 3 times wide area at least, you can see why it was probably some problematic issues. There was also some discussion that the genetics of the sterile flies may have become less effective at doing this. And so they've hopefully updated those genetics now. It does sound like they're being more effective now. So this is January of this past year. So again, now we're just focusing on Mexico. We can see that we've got pretty endemic status down in that neck here in 1,000 cases, active cases. The green here shows where the sterile flies are being released to try to help prevent it spread up into Texas. So this is, again, January 20th, 2026. A month later, things have moved north. We started doing sterile fly releases along the border as well. We're seeing an increase in cases down south. And again, I really just, I can't imagine what they're dealing with down in Central America with the extent of cases. I've heard the reports of wards of hospitals, being spilled out of the hallways with people waiting to be treated for screwworm on them. I've heard of people who have unfortunately died because, they got it up in their nose, didn't realize anything until it had actually eaten its way into the skull and into the brain of people. Just a very disgusting vermin. So this is May of this year. Again, we're getting closer and closer to the U.S. border. We're expanding those sterile fly releases into Texas. And then this is just the one from last week. Obviously, we've broken into the U.S. We're expanding northward. And then we've also seen cases that are getting more into the northwestern territories of Mexico as well. So again, basically, we're trying to keep it under control in the U.S. Unfortunately, Mexico and South and Central America, they're going to be in a hurt unit for the next 2 or 3 years until we can really start making a headway against this. This is off of the Texas Animal Health page showing the infested areas as well as surveillance zones. And again, this is just from last week. I just wanted to post that up there. This is just a screen capture of this map. It's actually 41 cases today. It went up over the weekend. Again, we're showing active cases. The reason it was kind of interesting as this developed, we weren't seeing very many cases at all on border counties in Texas. So we think there was a storm that blew a population of flies up the kind of that south central Texas area, and that's why we saw it start there versus seeing actual natural progression of the fly up across the border, because we didn't trap anything along the traps that we have along the border until we still haven't caught anything. So it's really interesting how that has occurred. And then this one, this was a podcast. If anyone's familiar with Radio Lab, this is a podcast back in March. He's talking about the return of the flesh eaters. Very interesting, especially if you're someone who's been paying attention to this for a long time. The sterile fly technique, which was developed by Dr. Edward Nippling. He was a farm kid in Texas. As in his formative years, he was the youngest kid, so he had the job of pulling screwworms out of cattle on their operation. So he hated this fly from his young age. So he helped develop the irradiation technique. And I recommend listening to this. They also had a panel of bioethicists talk about the morality and ethics of eliminating a population, an entire species. So they're also talking about different ways that we could eliminate this fly through gene knockouts, which could actually eradicate the entire species. And the panel kind of came to the conclusion that this New World screwworm, it doesn't really have any good use. It doesn't feed a certain frog or dragonfly that's like a main source. It doesn't control anything really important in the environment. So they said, really, in terms of a species that's a good candidate for us to just say, erase it from the universe, this is like the best candidate for it. And when you see what it does to animals and potentially humans, I got to say I'd be on board with that. And because we can preserve genetics too, so that if we ever find out that this fly has something that can cure cancer, we can bring it back if we had to. But yeah, that's an ethical consideration, and I just thought it was a very interesting, topic to think about. And that's what I've got. I know I kind of spent a lot more time on the New World screwworm, but I'm a cattle guy. You'll have to forgive me. Any questions I can try to answer for you? Yes, ma'am. Oh, so withdrawal time is when we use the medication. We need to know when are those animals going to be good to eat. So when we say we give an antibiotic, there's going to be that antibiotic within the tissues, basically the entire body. So we don't want to send that to slaughter and then have it be on the meat case if there's going to be antibiotics present in that food, because it's a human health concern potentially. And so they've done the studies on these to establish when those medications should be cleared from the system. So you'll have a meat withhold period. There's also a milk withhold period, depending on whether this is on a dairy animal or and this applies also to sheep and goats, of course, as well. So we have that, but we also sometimes have withdrawal periods for vaccines as well. Usually, I think that's more so because since they're going to be having reactive lymph nodes after a vaccination, they're going to go hang in a slaughter plant, and someone's going to say that that's an infected animal because it's got big lymph nodes. But why would you give a vaccination to an animal and then slaughter it 3 weeks later? It doesn't make economic sense. So, but yeah, that's what a withdrawal period is. Okay, thank you very much. Did I go horribly over time?
No, we're under time, so we are good. Any questions online at all? Am I missing any? Let me pull up here. Yeah. I see something in the chat. Let's see.
Oh, shoot.
Okay, so Dr. Pecker, she could see it. Were you guys able to see that presentation there by Dr. Galbraith? Kind of seeing some stuff in the chat here that you guys weren't. Okay, I'm getting some thumbs up there. Awesome. Again, awesome. You guys, this is something I think is very important that's coming up. I know there are some meetings and things coming with the state vet's office, Dr. Andrus's office as well. As he's going to talk about more of this in some other meetings that we have in some of our more private meetings. But from a public health perspective, I think a couple of things reiterating. I think this is people's livelihood that we're talking about with HPAI. You look at those numbers that Dr. Pecoraro and Dr. Jake had brought up, that there's millions of birds that are being depopulated. That's people's livelihood. They live off of that, and it's not an easy task to go and do any depopulation. I've never done it personally, but I've seen some operations of how that happens, and it's pretty intense. It's very working in PPE and working in hot environments inside these barns. It's long days, and like I said, we have seen some of this in our region here, the first district region, the 7 counties, but also in North Dakota, some of the HPAI, and then to kind of see some of this New World screwworm stuff. I'll try to get some of this information out to you guys so you can look more into it. But if you guys look at some pictures of what it looks like in humans and cattle, like what Dr. Jake and Dr. Pecoraro had in their slides, it's pretty nasty stuff. And like Dr. Jake said, I can't imagine what it's looking like down in Central America.
I would imagine, yes. I mean, I'm not a human doctor, but they say that in livestock it can be something as small as a tick bite that they will come to. And so you think about anything that just scraping your arm potentially, bite from a horse fly could potentially be enough. Scratching your nose, yep. Take a trip on the skin your knee or something, and that's all they need.
Yeah, this conversations I had with Dr. Jake before starting this. I even saw some of this getting brought up in like the Yellowstone episode. We did talk about some. They were mentioning New World Screw Room, but then they were also talking about what was it, foot and mouth or something, or foot, something like that. But yeah, they were mentioning it in Yellowstone and stuff. And I know there are a lot of people that do watch that. But again, and some of that high path AI thing, I know there are a lot of people out there that have backyard flocks. Some of the importance of that. That's why we're talking about this today. I myself, so our next speakers, we do have Dr. Weand. I apologize if I do get your name wrong, Dr. Logan, and then Ryan Herigstead with the Game and Fish Office. I've said in a lot of these high path AI things, but I don't hear too much ever, and this is nationally as well. Just what is the game and fish perspective of things, and working with with pheasants and with geese, like we've brought up here. Kind of talking about what the role is of the Game and Fish Office in something like this, and just to give some general detail into their operations and what some of their stuff looks like. I am a hunter personally, so I thought this would be an interesting topic to add on there. So I see Logan on here, and I think Ryan's with them. These guys are actually out at the fairgrounds. They are working out there right now, so they're not able to join us in person, but they are doing some of the family day stuff and some of their booths out, the Game and Fish stuff there. So are you guys able to take over here for a few minutes?
Hi there.
Oh, we had you there, and then we lost you.
Can you hear me?
Yes, we can hear you now.
Okay. Ryan's on a different meeting. I can see if I can have him jump on this for a minute, but if not, I can just take over. So if that works for you, we can do it that way.
Yes, that sounds good.
Okay. Let me just set up, and then I can I'll have to sign off and get back on a different screen.
Okay, we'll wait here for a minute. Well, let's take a 5-minute break. Let's get back here at about 10:12, and then we will reconvene while they set up.
All right, thanks.
Yeah, thank you. All right, everybody. I do have 10:12 here. We will reconvene once everybody gets settled here. And then just remember, there are some handouts and stuff over there on all this information. Some really good stuff. I got some from my colleagues here to talk about these things in more detail if you want some paperwork. And also my disclaimer, I am NDSU alumni, so go Herd. But they do have, so the SEER program that Dr. Galbraith was talking about, the team there, they have some really, really good brochures and posters and things that you want if you want to hang them around. I'll send some links and stuff out after this meeting. So you guys have that information and it's accessible to you. So now we are going to hear from the Game and Fish. Again, like I said, they are over at the state fair, so they won't have too long to talk with us here, but they do have a few minutes, and then we'll take some questions afterwards. So I will turn it over to them.
Hi, I'm Ryan Herigstead. I'm the wildlife health biologist with the North Dakota Game and Fish Department. We had started seeing mortality events in waterfowl, specifically snow geese and Canada geese in 2022 associated with HPAI. And every time we had a mortality event with more than 3 or 5 individuals, we were going out, collecting the carcasses, sending off the swabs, and every one of them for quite a while came back, positive. Anytime we had a suspect, it pretty well came back positive. And we saw, especially during fall migration, and then in the spring, we also saw, large numbers of mortality events where you'd have, these flocks with tens of thousands of geese in them. Every time they'd get up from a field from feeding or, roost water, there would be dozens left behind, sometimes hundreds. And over time, it seems like, those large mortality events have slowed down to the point where this last spring we just saw, individual geese here or there rather than the dozens and hundreds. So I'm not sure if, the ones that were more susceptible to it have already kind of been weeded out of the flock, so to speak, or if it's losing some of its pathogenicity or what's exactly going on there. But it is nice to see that the large mortality events seem to be slowing down that are associated with this outbreak. Anytime we do have, larger events, we still like to opportunistically swab and sample just to make sure the serotypes are the same and that the virus is not changing or it's, something else. But another issue we've ran into is the labs that are, responsible for doing the HPAI sampling are just completely inundated with samples from domestic poultry that unless we really, really need something sampled, like it's a species of high conservation concern, like a whooping crane or bald eagles, things of that nature, they seem to, kind of be deterring us from, submitting large numbers of samples just because they are already so backlogged. I'd also looked up a couple of studies. In 2023, there was a study in Washington where they were looking at hunting dogs specifically that were out hunting waterfowl and exposed to large numbers of birds that could have HPAI. And they sampled over 900 dogs. Only 4 of them tested positive for high-path antibodies. None of them had clinical symptoms. And yeah, they were all like waterfowl hunting dogs that retrieve large numbers of ducks and geese. So there is a certain level of exposure that's going on there, but it was, good to see that that was relatively low. So it's not an easy thing, I guess, for them to be inadvertently affected through, regular hunting activities. I think it was 2022, there was also a study in the Netherlands where they'd sampled over 3,000 dogs, and there was 13 or 14 with antibodies detected. There was one dog that had clinical symptoms, but that dog's owner also was clinical and tested positive for HPAI. So whether the owner got it, gave it to the dog, or if they co-got it from the same bird while hunting, that, we don't know exactly. But my takeaway from that is there is a small risk that's associated with waterfowl hunting, dogs, hunters, being exposed, but it's a relatively low risk. So we just encourage, education on the subject. Make sure that you're properly cooking and handling, the meat and proper carcass disposal just to prevent other animals from inadvertently contracting it and spreading it around. So if I was like myself as a waterfowl hunter, I am not overly concerned with it or my dog, but it is a known small risk. So that's just good to kind of get out to folks. I think Logan has some more information from this last spring, and then a couple anecdotal stories of, HPAI and the symptoms that were presented in a mammal. So I'll turn it over to him at this time.
Thanks, Ryan. That was great. Can everybody hear me?
Yep, we can hear you.
Okay, thanks. Yeah, thanks for having us. This is a great seminar here. I'll start out with a quick intro. So, yeah, I'm Dr. Logan Wayan. I'm the wildlife veterinarian for the North Dakota Game and Fish Department. I started my position here last October in 2025. And my background is actually in infectious and parasitic diseases of wild ungulates, particularly bighorn sheep and moose. So today I will share an update on what high path looked like in wild birds over the past year, species affected, and then, as Ryan mentioned, an anecdotal case in a mammal. So the time I started working at North Dakota in October 2025, that coincided with the fall migration, and so that means a lot of wild migratory birds are moving through the state. So during this time, we expect to see sickness and death in wild birds. So as an agency, we document reports from the public, and in certain situations, we may want to confirm high path through throat and cloacal swabbing, and that would be in a notable sized outbreak or in, as Ryan mentioned, species of higher concern. So this fall, we characterized the high path outbreak as overall mild. We received over 20 separate reports, so that means reports from the public in a different location um, of suspect high path in snow geese primarily. And those reports range from about one bird affected all the way up to approximately 300 in a given location. Um, and we did confirm high path through testing at two locations. As I mentioned, overall, these were primarily snow geese. Canada geese were also affected, but at a much lower level. And then the worst mortality events in snow geese were at Lake Richard in Sheridan County and Goose Lake. So in the spring, then we have the spring migration occurring. Um, the reports of suspect high path were fewer and at a lower severity than in the fall. And again, snow geese and Canada geese were the primary species affected in those reports. So through these reports, we also make sure to document the species affected in every case. And I just wanted to share just a list of birds in which we actually did confirm infections through testing of swabs. So for waterfowl, those included Canada geese and snow geese. And then we also had detections in bald eagles, great horned owls, and a peregrine falcon for birds of prey. And then also one detection in a turkey vulture. So those were the birds affected over the past year. And that's not, I should say that those were actually confirmed detections, not just suspect reports. But that then leads me to a anecdotal story that's important for this seminar because there's a public health aspect. Um, so as Heidi mentioned, uh, avian influenza can infect mammals as well. And in North Dakota, red fox has been a species we have had detections in for wild mammals. Um, and so back in April, on April 21st, we received a report from a game warden, actually in Minot, so very close to home, of a red fox pup that was found at the Minot disc golf course. And this red fox was reported to be having seizures and foaming at the mouth. And the report I got was that there were two young boys and a law enforcement officer that had actually had contact with this red fox. And so at that point, that is a concern given the neurologic signs, given the human exposures as well. And so our first step was to collect the fox, and then um, this animal, of course, then would have to be euthanized for testing because our first concern would be rabies, given that it had neurologic symptoms. So after that was those steps were done, we submitted the head to NDSU for rabies testing. And then another disease that wild mammals, especially the carnivores, can have is canine distemper. So our first two diseases we wanted to rule out were rabies and canine distemper virus. So fortunately, this fox was negative for both of those two viruses. Um, but after ruling that out, we still wanted to know what the cause of those neurologic symptoms were. So we decided to test for high path avian influenza because it could fit with what we were seeing. And sure enough, the red fox was positive for high path avian influenza. So thanks to the North Dakota Department of Health and Human Services and NDSU pathologists, we were able to get a definitive diagnosis on that case and then communicate health recommendations with the public throughout the incident. So I just wanted to share that because it does hit close to home here in Minot too. And just to spread awareness that there is a risk to handling and contacting wild animals, whether that's rabies, avian influenza, other diseases. And so, again, I wanted to thank Zach for hosting this seminar, and thanks for the opportunity for Ryan and I to share some information from Game and Fish.
Yeah, thank you guys very much. Thanks, Logan, and thanks, Ryan. I know, does anybody have any questions? I know there was kind of one earlier. Anything online or in person here? Nope. Good. I got a couple questions here. Jake, go ahead.
I was just gonna ask the question on, so for hunters who are taking, Canada geese this fall, in order to try to minimize the potential for bringing something back to their own homes, their operations, how does field dressing work? I'm not familiar with how, it goes with waterfowl in terms of, the document, what you got. So what are the recommendations for like trying to do any field dressing, in order to prevent bringing something home to a commercial or a backyard farm?
Yeah, for field dressing and that you have to follow obviously the migratory game bird laws where you have to leave at least one fully feathered head or wing attached to the carcass. But I would just recommend, good sanitary procedures, wear disposable latex or nitrile gloves, and wash your hands thoroughly after handling the birds.
Great. Thank you for that question. Lisa, did you?
My question just touches on testing. It's been mentioned a couple of times that it's looked at rabies first, and if it's not rabies, then this is tested for. Is that common practice? Like if we send it to Health and Human Service lab, the micro lab, the state micro lab for rabies testing, if we send a cat or dog down there and it's negative for rabies, is it automatically tested for HPAI? Or anything else for that matter?
Yeah, that's a good question. Heidi, I see you as well. Do you want, you can go for it.
Yeah, so if there are neurologic signs in any animal, typically they often come to us, especially if it's a large animal or wildlife rather than the state lab. And us, I mean the veterinary diagnostic lab in Fargo. And then we will do, what's different about our test is we have veterinary pathologists on site who we do the test for rabies, but then we look at the tissues. So then we as pathologists are trained to see if there's inflammation or any other thing that might give us a clue what's going on. And that's usually when we're talking with Logan or with Dr. Andrews out of the state, vet office in terms of what testing's done next. HPAI is, like I said before, we don't just get to do that test willy-nilly. It's a regulatory test. So we have to get permissions and also see who's going to pay for it. So that's typically how that happens. But as pathologists, we're trained to look for other diseases. And so we have clues in what are the next steps in diagnostics would be, if that helps.
Thanks, Heidi, too. And I'll just follow up that this fox, as Heidi mentioned, we're looking for lesions and pathology. And so this fox did have encephalitis and also, so that's inflammation of the brain and then also inflammation of the heart and liver. And so that was also another, those were lesions that could fit with avian influenza.
Yeah, and I was going to say I really appreciate you bringing that up, Logan, because we do have a rabies program here at First District, and so I guess I didn't really realize that it kind of looked very similar to what he was describing, like a rabies symptom with foaming at the mouth, and some of those symptoms very similar to rabies. I guess I didn't realize that, so that's good information. Does anybody else have any questions for Game and Fish before we move into Health and Human Services?
That comes back negative, then you follow the process of elimination, then you look at that HPAI.
Yes. Were you guys able to hear that?
No, I did not.
If it presents with rabies symptoms like the neurological, the foaming at the mouth and things like that,
and it's tested, it's negative, I'm assuming through process of elimination, that came back negative, then move forward with HPAI, correct?
Yes, if there's other consistent lesions that would make us suspect HPAI. Usually it's rabies and then distemper are the top two to immediately rule out. And then we'd continue with other testing if required after that.
Got it. Thanks.
Anybody else? All right, again, I appreciate that. Thank you guys very much for taking your time to speak on some of these things. It's very insightful to us here. So again, thank you for that. We're now going to move into, I have Levi Schlosser. He's one of the epidemiologists at Health and Human Services. He's going to kind of look at this and talk more about the human side of things and what you look for. Again, it's kind of just to reiterate some of the things that we would look at from the human health perspective as to why this is so important as well. Again, we're talking about that reassortment of the gene and of the disease itself. If it does develop into something, widespread that people are afraid of, kind of like a COVID thing. Obviously, we don't want to bring that up and get that anywhere, but there is a possibility, just like anything else, that something could mutate and it does start doing some human to human transmission. So, Levi, I appreciate you being here today. Are you able to pull your slides up?
Yes, I can. Let me share my screen here. Are you guys able to see my slides?
Awesome. We see it.
Perfect.
All right. Thanks a lot.
Thank you. I am just going to do this. My name is Levi Schlosser. I am our respiratory disease epidemiologist here with the North Dakota Department of Health and Human Services. And I'm just going to give an overview of what we do on the human health side of the response for avian influenza. And so I'm sure you guys are getting well versed in avian influenza, but typically when we say avian influenza or avian flu or H5, this refers to a type of influenza virus caused by influenza A viruses that typically infect birds or avians. These typically spread naturally among wild birds, as you've heard, and they can infect domestic poultry and other animal species, including humans. Typically, these viruses don't infect humans. However, there have been sporadic cases of human infections with bird flu viruses that have occurred in North Dakota and worldwide. Since 1997, most of these infections have been reported from H5N1 viruses reported from 24 countries. The severity of their illness really depends. It can range from mild to severe, although most of the cases have been fatal, about 50% of known human cases. And beginning in 2022, we've seen this outbreak spread to the United States across domestic and wild poultry, and then again through dairy cattle beginning in March of 2024. We describe these viruses as highly pathogenic because it does cause such severe morbidity and mortality in birds. And typically, these viruses are spreading through a few different ways. Direct contact between infected birds and other animals is going to be the most common source of spread from contact with their environments that have been contaminated by the virus. So bedding, coops, any supplies that those animals have or may have that virus come in contact with can be contaminated and be potential ways for that virus to spread to other animals. And also through fomites, such as dust or droplets through the air, typically can cause a wide range of symptoms that would resemble normal or what we would expect to be regular respiratory symptoms for influenza, including fever, cough, sore throat, runny nose, shortness of breath. The HPAI symptom that is most common in humans that we've noticed has been this eye tearing or redness or conjunctivitis. That's the most common symptom in humans that we've noticed, although any of these might occur and be more non-specific. Because it can have such a wide range of symptoms, mild complications can occur, including really high fever, shortness of breath, altered consciousness, or seizures, and that can cause complications like pneumonia, respiratory failure, and even death including the other conditions listed here. Now, overall, the Centers for Disease Control and Prevention assess the overall risk to the general U.S. population as low for avian influenza. However, that risk is a little bit higher for populations that are in constant contact with potentially infected animals or their surfaces or fluids. So those individuals who are most at work are going to be typically those farm workers, animal handlers, individuals who raise poultry are going to be are at increased risk because they have more prolonged, more intense contact. So their risk group is a little bit higher. Typically, these viruses spread naturally among wild aquatic birds worldwide and can affect domestic poultry and other bird and animal species. They don't typically infect humans, and so that's why the general risk is low. Although sporadic human infections have occurred, the risk is low, but it's not zero. And just looking at that picture, since the start of 2024, there has not been any known person-to-person spread at this time. The general risk is low. We've had a total of 71 cases and 2 deaths since the beginning of 2024. Looking at the spread of where those cases have been in poultry, and specifically domestic and backyard poultry, we can see here this heat map kind of shows that it's concentrated in certain areas of the Northeast as well as in the Northwest parts of the country. Really, we see H5N1 hit birds in, I believe, almost every jurisdiction in the United States, all 50 states and 1 territory. To provide a little bit of context on the size of the U.S. poultry flock, there's more than 378 million egg-laying chickens in the United States. And in 2023, there's been more than 9.4 billion broiler chickens and 218 million turkeys were processed in the United States. And so 206.41 million birds have been affected across the start of this outbreak, including over 1,100 backyard and domestic flocks. When we look at commercial poultry, this is slightly fewer flocks, 1,012 different poultry outbreaks, but that's where we see a lot of the numbers come from, are these large commercial operations. We can see that those are concentrated a little bit more commonly in the Midwest, Minnesota, South Dakota, and including some in California. And then these are charts that I believe we've seen already, states with HPAI detections in wild birds. North Dakota's had 538 detections in wild birds since December 30th of 2021. And in dairy cattle, we've had over 1,000 heads of cattle, I should say, impacted by avian flu. Now, I should say the majority of these cases have been impacting West Coast states, primarily California. In North Dakota, we have not had any dairy cattle test positive for H5. And this is a chart I believe Dr. Pecoraro showed, showing detections of HPAI in wild and captive mammals, including some wild canines in the state. And finally, looking at the current situation for human cases, again, 71 total cases. A majority of those are following exposure to infected dairy herds, while with the remainders occurring following exposure to poultry farms or culling operations. The remaining cases have either been undetermined as far as the source, or they've had an unknown or other animal exposure. There was one additional case that was previously detected in a poultry worker way back in 2022. This first death reported in the United States was in Louisiana. But looking at North Dakota specifically, we've had a total of 53 detections since the start of this outbreak in 2022. Looking at this year, we've had 4 so far reported to us in 2026. We've had a total of 180 individuals monitored following their exposure, including 16 individuals who went on to develop symptoms and be tested. 7 of those were actually tested for avian influenza, and all of those were negative. Looking at the breakdown of where we've seen a majority of those detections occur, a majority of those have been in backyard domestic flocks or poultry operations that are non-commercial, 14 commercial turkey operations, and over 612,000 birds since the start of our monitoring. Now, these are kind of spread across the state as we have had multiple flyways as those birds cross through North Dakota. It can occur in really any county, and there isn't a rhyme or reason to it, although we have seen the majority of those occur so far in Lemoore and Dickey counties. And now when I say monitoring, what I mean is the public health action that we take on the Health and Human Services side to prevent the spread of avian influenza. The Centers for Disease Control and Prevention have multiple systems in place that we really use year round to monitor key flu trends. This is not just limited to avian flu. These are also for human influenza and other respiratory viruses. So that includes case reporting, public health lab monitoring, so monitoring the overall testing volume that our public health laboratories are doing each day and each month. Looking at the clinical lab trends, so where our hospitals and our healthcare providers are testing, looking at emergency departments, so who's going to the emergency department for fever, cough, shortness of breath, or flu-like symptoms, or who's going to the ER and mentioning exposure to poultry or birds. And then finally, wastewater surveillance can give us an idea of when there is virus detected in a community that can sometimes indicate that there is potential transmission occurring either in the water runoff or within humans. And where we come in on the Health and Human Service side is primarily the case reporting. When there are exposures that occur to infected animals, we follow up with anyone who is exposed to those animals to monitor them for a period of 10 days. Typically 10 days is when we've seen the duration of symptoms last. Past that 10-day mark, if you are symptom-free, we typically will discontinue monitoring and just encourage proper PPE following that. If you go on to develop symptoms during that 10-day period, then we can coordinate testing, and we also assist with overall flu surveillance at the state level. And again, because the highest risk to North Dakota populations are those who work directly with infected animals, our main recommendations are going to be regarding the risk to farm workers who are handling these animals, what PPE they're wearing, recommending that they take antiviral treatment and chemoprophylaxis if they do go on to develop symptoms, making sure that they receive their seasonal influenza vaccine. They have not already done so. Raw milk consumption is something that we recommend they avoid. And overall respiratory wellness in general is going to be really helpful for cutting down on not just on the spread of avian flu, but other respiratory viruses that we know can circulate, including regular seasonal influenza. Again, the most common symptoms that we've seen nationwide have been fever, cough, shortness of breath, sore throat, runny nose. Some unusual symptoms can occur, including nausea, vomiting, diarrhea, things that we would normally associate with influenza, but conjunctivitis has been the most consistent symptom that we've seen for this virus. And so that's something that we really encourage people to keep an eye out for, as that can help give us an idea early on of potential infection. And so what we do is we monitor these individuals who are potentially exposed. We set up either daily, semi-daily, or regular check-ins with those individuals to check in on their health, make sure they're monitoring their symptoms directly, and then provide education for them if they have any questions about what symptoms to watch out for, how best to prevent the risk to themselves and others, what steps they can take to protect themselves and their flock or their animals at home. And then we collaborate really closely with Department of Education, or I'm sorry, Department of Agriculture and USDA, Fish and Wildlife. All these programs are going to be typically taking the lead on the animal side, and then we'll provide those recommendations on the human side. And then finally, if those individuals do need or go on to develop symptoms, we'll help coordinate getting them tested and then getting those results to them in a timely manner. Typically, the people who are at most risk are going to be, again, those poultry producers, staff, or anyone who has contact with those birds or their environment. Often we see it's not just the owner themselves, it's sometimes them and their their family, their wife, their kids that go in to pick up the chickens, hug the chickens, feed the chickens, and those are going to be potential risks for everyone to be exposed to those viruses, even if the animal seems well appearing at the time that they interact with them. We also work with state officials who have had contact with those infected birds, or any responders that will travel outside North Dakota to assist with other states' avian flu responses. When they return to North Dakota, will finish out their 10-day monitoring period once they arrive back in the state. And this just goes to reiterate that farmers and farm workers are at the most risk for avian flu infection. Most of the individuals who have gone on to develop confirmed H5N1 virus infection have actually had exposures associated with either commercial egg or poultry. Most of those, again, were dairy cattle, and 24 were poultry farms or culling operations. The remaining 6 cases shared exposure either with a backyard poultry source or their exposure source was unknown. Sometimes those workers get identified before any animals on the property end up testing positive for H5. And so that doesn't necessarily indicate that the virus is spreading from people to animals, but it is indicating that it can happen, and sometimes you might not notice it in the animals before you go on to develop symptoms yourself. So that's why it's really important that we take the steps to protect ourselves, as well as we take those steps to protect our animals. We do have some resources available for producers. We do have instructional materials on how to safely don and doff PPE, resources for producers who are impacted. These flocks can be devastated often by these viruses, or they're very high morbidity and mortality when it does come to the backyard poultry in particular. And so we do have some resources available to help those individuals who are impacted, because it can be really devastating and a really tragic event for them. And we've also got some informational materials available on our website. We've got some fact sheets that we can send out by mail or by email that kind of details all this info. It's got a temperature log to help them with daily symptom check-ins. It's also got our contact info. If they go on to have any additional questions, they can always reach out to us here on the stateside. And as far as what we recommend for next steps, really, because these viruses can cause severe illness and death, it's important for anyone with those exposures to infected or potentially infected animals to take any recommended steps to protect their health. There has a recent CDC report recently summarized how influenza A viruses can affect the GI tract of mammals, including people, which really recharacterizes how the symptoms that we normally would expect for influenza can sometimes not be present. We'll see more of these GI symptoms present where we normally wouldn't. One key question that we've had has been the potential health threat to people from eating or drinking either food or beverages that contaminate with those viruses. So raw or contaminated unpasteurized milk has been a really common question that we're currently doing more research on, but overall the risk to the public is low at this time. And so finally, I just wanted to give my contact info here. I've got some also links for respiratory illness. If you do have more or are looking for more information on regular seasonal flu, RSV, or COVID-19 right now, typically July is where we would consider the off season for influenza and other respiratory viruses. But typically we'll start tracking for the new season already in August. We'll start seeing those pick up in September, October. But otherwise, I will be on for questions if anybody has some. Thank you for your time.
Awesome. Thank you, Levi. Really great info. Does anybody have any questions here in person or online? I know a couple of things to kind of reiterate what Levi was saying was from the public health perspective, like you mentioned, that the CDC, it is deemed low risk to humans overall. Obviously, that increases if you're working with poultry day in and day out, if you're messing with your chickens, like the kids, running out and playing with them. It's a perfect segue into kind of what our next section is going to be with more PPE and kind of some tips that you can do if you do want to handle the birds and you are closer to them. So Yes, so this is where the last hour of our seminar here. We are going to have an online. We're going to have a presentation here, a slideshow from my colleague Crystal Coons. She is in my role down in Bismarck, Burley, and covers those counties down there. She's a nurse, so she's really good with some of this PPE stuff, and I'm going to let her take over from here. And then once she's finished with that, we'll do our hands-on stuff, and that's for the online people. That's where it'll kind of finish up on that end. Again, thank you guys for joining us. I hope you guys are taking away some good information here and some things that you guys can do going forward and doing this in your own communities. If there are some things that you're more interested in, or you do have some questions on things, or need any connections or anything like that, please reach out. So I am going to turn it over to Crystal in here. I'm going to pull her slideshow up. Yeah.
Before we're done, just let them know that if there's anyone who wants to have, come and do those presentations we've been doing for community events,
Sure.
you can always get in touch with me and set something up.
Okay. Yeah, online, Dr. Galbraith here with NDSU Extension, he did say that if anybody in your communities would like any more information on some of that HPAI AI he was talking about, certainly to reach out to him. We can get you his information and set some of that up, and we can bring it out into the counties and across the state. I have one more question here. Yeah, absolutely.
That's usually the state vet office that's coordinating that.
Favorite people out there.
Yeah, so you might get a little bit of some assistance. So usually the way they do that in poultry operations, it's a CO2 or the carbon dioxide or carbon monoxide, but also nitrogen, it's a pollen that goes over the top of the birds, and they have basically this application. I've seen the videos. It's not a pleasant thing to have to think about, but it is relatively, it's pretty low stress. It's very compliant. There's not a lot of, they basically, CO2 levels come up, they fall asleep. It's not a violent or comfortable thing. No, because you're talking about in some of those operations, even in a state of 60,000 individuals, you have to get
Was built into the presentation, so I actually did.
Unfortunately, when this first started, they were doing it in other states. ESD or ESD plus, which ventilation shut down, plus heat, they basically shut off the ventilation system. That is not, that's not acceptable. That's a bad thing. Whereas with this foam, it basically knocks out birds. And Nate Beam in the vet office, he showed me some pictures. He's told me about this. He helps a lot of those. And the worst that he's had happen is that the foam comes in and it's like it slowly rolls over the top of them. So it doesn't scare them. Because there was one time a turkey put its head up through the foam, and all he had to do was just literally just push it down. There was no resistance, no stress in response to it. So I haven't seen one personally. I had the opportunity in January of the outbreak to potentially have seen one, but I thought that it wouldn't be a good idea for me to go to do a depopulation of an infected flock, because that evening I was doing one of our presentations with producers. Send a vet message for me to go to an infected flock and then come talk to a bunch of backyard producers about biosecurity. Otherwise, I would have gone in there. I want to be a part of one, but that one just didn't work out.
For the online crew here, we were kind of talking. There was a question of the group of what depopulation looks like. So Dr. Galbraith was given some more information on that. I do know NDSU Extension does offer a depopulation class type of seminar. It's a 2-day thing. I don't know if they're still doing that with some funding and stuff, but it is a really good course if you guys are able to take that, get out there. It was in Carrington the last time that I did it with one of my colleagues here. They kind of go into more of that and how that's done. I do know in North Dakota that when there are some depopulations happening, it is private contractors and stuff that come in from out of state and are set up to do this kind of thing. Typically, it involves the foaming where they close the hen houses and the barns, and then they do extinguish and put this foam in there, kind of like a fire extinguisher type of thing, but it's a foam that builds up and slowly does kind of knock the birds dead. And then obviously there's a lot of the pickup and cleaning them, picking them up, and then letting them decay appropriately, which involves a whole process that I don't know the science to, and getting to certain temperatures and rotating them in piles and things like that. And then I know from other discussions with the state vet and things, there are some other techniques where it does involve like the turning up, closing the vents and everything, and then slowly suffocating them with the heat, which in my opinion as well, it seems a little harsher than everything. So I don't think I would advocate for that either, but it is something that can be used and be done. So I'll just throw that out there. And then, yeah, so now we're going to have Crystal in, like I said, she's going to go into some of this PPE and what you can do personally. And then we're going to give people here an opportunity to try some suits on if they want to. And you can actually experience and see what it feels like, even in a controlled environment, what it feels like to put on these Tyvek suits and put all this gear on and wear it. And then work in those conditions on top of it. So being mindful that we are in a controlled room, you're not working out in 85, 90 degree weather, letting something like this happen, and then going and picking up thousands at a time, so I'm going to turn it over to Crystalline.
All right, Zach, thanks for having me. Again, I am from the Southwest Central, the state response coordinator. So basically just the county south of what Zach takes care of. I'm going to focus specifically on PPE today. We heard great information about avian influenza from all of the experts in the room. So I am really not going to touch on that. This picture here is of my own backyard flock. And we have been blessed with 8 wonderful egg-laying chickens. So personal protective equipment for avian influenza, the CDC does have a couple different levels. They have a medium exposure and then a high exposure level of the PPE if you would be interacting with birds or cattle or anything that would have avian influenza exposure. The biggest thing with PPE is the donning and doffing process. You have to put it on in a certain order and you have to take it off in a certain order to prevent self-contamination. And we will go through all of those steps today. So medium exposure settings, this is a flyer directly from the CDC, and it didn't specifically tell me what medium exposure is. Unfortunately, I don't know. But if you are in a medium exposure setting, there are 4 types of PPE that they want you to wear, and that is a NIOSH approved respirator, safety goggles, gloves, and then outer work gloves. And then it goes into how to remove those as well. You would remove your outer work gloves, your disposable gloves, your goggles, your respirator, and then you want to wash your hands. And that's for a medium exposure. And then we go into high exposure. Now, high exposure is going to be if the animals are confirmed. You know that they are showing symptoms. Maybe you have to depopulate going into that exposure. So this is more full PPE. You're going to cover your hair or your head with a head cover. Some of the Tyvek suits just have a hood that can come up as well. You're going to be wearing safety goggles. You can have an optional face shield that goes over the top of those goggles. You are going to have a NIOSH approved respirator, an N95 mask. You're going to have coveralls that keep you dry, and you could have an optional waterproof apron over the top of those coveralls. You want disposable gloves with an optional outer work glove, and then you want boot covers or actual boots, disposable boots. And it's just it's going into there. So before we get into the actual process of putting on the PPE and taking off the PPE, I wanted to talk just a quick minute about what a NIOSH approved N95 respirator is. What that is and it's approved mask that's saying it's going to keep out the particulates at a high enough rate that you shouldn't get illness when you're wearing that respirator. We have different kinds, 3M makes different kinds. It will say that right on the box, NIOSH approved N95 respirator. With those respirators, though, they only work as good as the user. So we need to know how to put them on, we need to know how to take them off, and we need to have what's called a fit test. A fit test is a procedure that I think Zach can do it here at the health unit. If you are interested in knowing if that mask fits you correctly, we can either use a port count machine or we can just use a bitter spray in making sure that that mask fits your face correctly, seals your face, and is preventing those particulates from getting in and out. Any questions on that fit test before I move on?
say if it's dust particles or something like that, depending on the size.
There is, but it is, according to this, it wants you to have the NIOSH approved N95, so that would be up to the medical grade. You wouldn't want to make sure that it's a dust particular. You,
You have them separated.
and then again, when you are fit tested, you want to make sure that you know how to put that mask on and off. So I'm just going to touch a little bit on a user seal check. So when you're trained to put on your N95 mask, you would do a positive and a negative pressure check with your mask on. So once you have it on, you blow, make sure you get a good seal. You blow out real hard. Do I feel any air leaking? You suck in real hard. Do I feel any air coming in, any seepage anywhere? And that is, after you have been fit tested to your mask, that is your check out in the field to know if you got that mask on correctly, that positive and negative pressure seal check. And there is handouts on all of this stuff back there too. So now we're going to jump into the donning and doffing. So donning is putting on the PPE and doffing is taking off the PPE. And for my people online, if you guys are still hanging on there, I think what we'll do is we'll watch both of the videos and then we'll do the hands-on. So we'll go ahead and just get started with that first video. Is this touchscreen?
No, it's not. Nope.
So as a nurse too, I do have to say I'm sorry, but I put this at the highest level of PPE that's available out there. So I picked Ebola. So we are looking at the highest level of PPE. You can pick or choose if some of this stuff isn't fitting for you and you don't need that extra level, that's fine. But I needed to make sure that you guys were trained in the highest level. All right, it's playing here, but I'm not hearing anything.
Yeah. Sorry about that. I did not test the audio for videos prior to this meeting. Let me see here. Any ideas, Brandon? I know we talked about this.
So a high level situation like this, say it's Ebola, how does one go about even getting a drink of water in that situation?
No, when you have your PPE on, you're limited to a certain amount in the patient's room, a certain amount of time. And you should always be directly observed, making sure that there isn't a breach of your PPE. And then when you come out of that area, you're also observed.
What about people who are responding over in African countries, like working on the ground? And that's just. You only work for 2 hours at a time, have to go off and then drink some water, do your business, and then go through it all again multiple times a day.
Yeah, and it's very fatiguing to be working in full PPE. So, 2 hours is truly, I guess I'm not sure what their actual time limit is over there on the ground, but anything more than 2 hours, even like in the United States, if they would be responding to that. Even if they wear a PAPR, which is breathing the air, it's kind of the matters can help you still limited.
Everybody online, we're just kind of troubleshooting the audio here quick. Again, I apologize. I did not test the videos prior to this. I appreciate Chris bringing up the fit testing here. So for fit testing, this is something that we do for our public health unit here. But if there's anybody in the public or anybody on this call that is an online, that does have like an online or that does have a backyard flock or anything private, or you have a family member that does, this fit testing can occur at one of your medical facilities. The only place in the Minot area is the Sanford Health Clinic off of Highway 2. If you have any more questions on that or anything, please let me know. Again, these N95s that Crystal Lynn is talking about are the medical grade ones. These are ones that we would use for our response to any infectious diseases. If they are like an Ebola or something like that, we would maybe change a little bit and add some more PPE to it. But again, this is the number one thing you can do to prevent some of the spread of disease is be trained appropriately in PPE and wearing it and taking it as serious every time you don and off, taking it as serious as you are being exposed to something like an Ebola, and training that way so that you pay attention to everything that you're doing very closely. I do know, like in Ebola, when that happened back in 2014, the outbreak down in Texas, the nurses, the people that were again exposed to that, they became exposed in the after action report accordingly to the virus as they were coughing. So being conscientious about that.
This speaker up here.
Okay.
And then okay. Try it now.
Just bear with us for. Oh, never mind. Skip the video. All right. Can everybody see this video online? You're not seeing it?
No.
So I should be screen sharing. Well, all right. Can everybody online see the video? Okay. And then I'm going to play it now. Let's see if you guys can hear it. I can't hear it. Okay.
They just want to click, the links were provided in the PowerPoint, so if they just want to watch it on their own, because really at the end, we're going to be doing
Doing it.
hands-on, so.
Sure. Okay, we'll do that. Everybody online, some technical difficulties here. So what we're gonna do is the link is in these slideshows, and I'll get this sent out to you guys once you to get you to watch it. If you'd like to see it, we are going to do the online piece. Do you have any more on your slideshow for anything? Okay. Okay. So I will stop that there. All right, everybody. So this is the online portion of the seminar. We're going to transition into that here in person. You can feel free to go. It's going to be a lot of the background noise and people suiting up a little bit. Again, I do appreciate all of you joining today. I hope the information was insightful and I hope you took something from it. Please let me know if you do have any questions or need any connections to anybody. Again, a very important topic, both of these topics with HPAI and some of the New World Screwworm stuff that's coming up. We are remaining vigilant and watching both of these to prevent any disease transmission. So again, thank you guys very much, and I hope you guys have a great rest of the week. And thank you to everybody that were speakers on here. I will be sending some stuff in the mail to you guys. So thank you very much.